I think we are done with the viruses, right? Last time it was complete. You remember?
Yes.
We finished it.
Yeah.
Yeah.
All right. So I would say that when you go over that chapter again, you guys can watch a lot of videos. I showed you, I guess, last time some videos. 1 second. If any of you have taken anatomy and physiology, I think it's just the repetition of these 2 chapters. Any of you have taken A and P?
I've taken it.
I'm taking it right now.
Oh, okay. Yeah. So did you reach innate, like adaptive immune system, innate immune system there or no? You guys covered it?
I don't think so, not yet.
Oh, okay. All right. So these 2 chapters, I feel like every chapter is very interesting chapter. Everything is like kind of new. And that kind of keep us engaged into these, learning modules. And these 2 chapters, chapter 14 and 15, although these are chapters, we go slide by slide. But let me tell you that if we look at our immune system, how it works, I think it's a pretty good understanding in these 2 chapters that when we get sick, what are different types of cells in our body, how our skin protects us. It's like a first aid box. And like, when we go to the doctor, like when there are mild symptoms, we get flu, and let's say we are really not feeling that well, only then we go to the doctor. Otherwise, we feel like, oh, it's flu. Within 15 days, I think we will be okay, because how our immune system works. But if it goes beyond that comfort level, we go to the PCP. And PCP has only that much authority, or you can say like to deal with our symptoms. They can prescribe some like Tamiflu, or they will say, sometimes they say, oh, you are late with the flu and we cannot give you anything because virus will go itself away. Just take medicine for fever, take rest and plenty of fluids, that sort of thing, right? That's what they do normally. But sometimes, like if we have some kind of a chronic disease or something that PCP cannot handle, we go to the specialist. Like, for heart problems, we go to the cardiologist. For skin problems that PCP cannot handle, we go to the dermatologist. For other kind of, we have internal medicine for ENT, eye, nose, and throat, those specialized doctors. What I want to say here is that our innate immune system is just like our PCP or our first aid box, that it will act on any infectious pathogen or same way. They don't discriminate between a virus and bacteria. Kind of, they like, the moment we get a small cut, we go to the first aid box, take any bandage and apply it. That's how it works, the innate immune system. But if you look at the adaptive immune system, that's where your specialized or specialization, special skill of the doctor comes into play, that they have a particular skill of these cells, our adaptive immune system, that they are going to take care of one. If it's bacteria, they are going to work differently. If it's a virus, they are going to work differently. Like T cells work differently as compared to the B cells. So we will talk about that in chapter 15. Now, one more thing I want to tell you, that it's not like the way we are going to talk like slide by slide, or like this system is coming into play. It's like sometimes, when we get the infection, sometimes, not sometimes, but all the time, I think definitely our innate immune system comes first. But later you will notice, after you complete chapter 14 and 15, you will notice that, oh, everybody is working like at once to keep the pathogen low in numbers, so that they don't reach that threshold and cause more damage to the host. So let's see what it is. So do you guys know what innate immune system means, or what does innate means here?
Is it already like built in?
Yeah, we are built in with this immune system, right? Now, always keep one example in your mind when you're looking at the innate immune system. A little baby is born, right? Now, tell me, we are talking about the innate immune system. The moment baby comes into this world, what the baby is born with this innate immune system. Let's understand it. Let's discuss it. So tell me what the baby has that is going to protect him or her from the pathogens, because in the hospital you can imagine how many bacteria are going to be there around the baby. What is protecting a baby, the newborn child?
You said what is protecting them? Like once they're born?
Say it again.
You're saying like once they're born or like while they're in the womb?
No, I'm saying that what is that, like what part of their body or immune system is protecting them at that moment, what they are born with? Because Jasmine said that
the Virnex?
Huh?
The Virnex that they're born with, like that protects them?
Is it the antibodies from their mothers that they inherited from their mothers?
Mm-hmm. Antibodies from mother. Okay. What else?
The secretion that they're born with on their skin, the Virnex, that helps as well.
Okay. Like any example of any body secretions?
Well, I know that the Virnex is to like help maintain their body temperature when they're coming out. But then also, babies don't sweat really, do they?
not at the moment.
I'm like, I don't think they sweat that early on.
Yeah, but you're right. Like there are some, but definitely saliva is there. I think babies do produce saliva, and yes, you're right that body secretions, but not the baby. What else? What do you think? Because they are landing really in the hospital, and you can imagine, like even we as a like nurses or anybody around, we are really not that. We are not sterile, right? Sterile. So you can imagine. So yeah, that's how I'm saying that you have to keep that example, that if you look at the baby, I think the biggest part or the largest part of that baby is the skin. Okay. So skin is a beautiful system that is going to keep most of the microbes away. Because, you can look at our skin, it's very tightly woven, cells. And even if you look at your skin, like, every day we are taking shower, most of the like outside pathogens or anything that is coming to our skin will be washed off. Secretions like sweat you're talking about, the oil you're talking about, they are going to take care of all those external bacteria, and they may be not all the bacteria who are coming or landing on our skin or on our body tissues, they are not pathogenic, but we really like, even they don't like to sit on our skin because they are not going to get much out of it. But yeah, there may be a potential pathogen, and that's what we are going to talk about, that how our immune system comes into play. So these are the guidelines, or you can say this is, some people say that, oh, is there any study guide? So objectives are actually our study guides, that where we talk about that, you can summarize in the end, what are the basic defense system, what are the 3 lines of defense system, we will talk about that in a minute, and the nature of innate immune system, that is it specific, is it non-specific, what happens in it, what are different organ systems, the cells or the secretions that make our innate immune system. We are going to learn about the receptors and, pathogen, recognition patterns. We call it PRR, or we will talk about that in a minute, or PAMP. So what they are, PAMP, these are new terms you will learn. Pathogen recognition receptors, what they are. We have to talk about the composition of blood, that what our blood is made up of, and how blood also takes care of our these pathogens or infections. What are those cells? They fight the infection. We are going to talk about like white blood cells. Then there are our B cells, our T cells. There are macrophages, all sort of things. Also, those are also white blood cells. So we have to know our blood, and how white blood cells like take care of these pathogens. A little bit about the lymphatic system, and then a little bit more in detail about the processes, like how inflammation happens, or what are different chemicals they are secreted, like chemokines, cytokines, and how phagocytes, like cells in the blood, the moment they see any pathogen, they are going to phagocytize it, means it's like an endocytosis process, and they engulf and take care of it. What are those cells? And we are going to talk about some other chemicals like interferons, the fever, the complement system, and how it works. So that's a part of our innate immune system. So let's first understand what innate immune system is. And like Jasmine said, that it's something that we are built with, or all elements. It's very important that the moment baby is born, what is she born or what is he born with? All those elements, we call that as an innate immune system. Always remember that your innate immune system never keeps any memory. Like your B cells or T cells, they, like the moment any pathogen visits our body, they keep the memory. Memory means until we die, we do have those cells, and they know like, oh, you have visited me when I was 2 years old. And if the same very pathogen visits again at my 50s, those cells will come forward and they will take care of that pathogen. So in innate immune system, that kind of thing doesn't work. It works only with the adaptive immune system. When we adapt to something, that like in the second time, they come very powerful attack on the same pathogen. Available on short notice. So that's a good thing, that the moment we are attacked by these pathogens or any external kind of microbes, they will be taken care by the innate immune system. And there are a variety of tools that innate immune system is made up of, that they use one at a time, the second, or like in a different ways, and they can tackle the problem. They can solve the problem. But always remember, that's very important. I can ask you this question: do they have memory? No, they don't keep any memory. They just work like irrespective of who is visiting our body. They just take care of it, but they never remember that they visited them earlier too, and they're non-specific. So what are the examples, what they are made up of? So if you can imagine, like if you look at your body, look at the lining, like our lining starts, the inner lining of our body, we have outer part of our skin and the secretions, right? That is the first line of defense, along with the, look at the mouth parts, the esophagus, the stomach lining, the intestinal and reproductive structures, then anal structures, like all those linings. It's just like a tunnel starting from mouth to the anus, and that lining also protects us. So we are born with, the baby is born with that lining. And what's in that lining? Like, if you look at the intestine, that lining is replaced every other like 3 months, because those cells, they die, and maybe they are not good. And in that way, they also get rid of other pathogens or any other external microbes. So physical barriers, that's what we are born with: the skin, the mucous membranes, because the membranes in this lining, they produce the mucus, and mucus is good to take care. Like when we cough, if you look at coughing, that mucus takes care of the pathogens. These phagocytic cells, the natural killer cells, that's what we are born with, that they are present in our bodily tissues as well as in the blood. So we have blood, the baby is born with the blood. There are some proteins, like, that play a role in complement system. There are some other interferons, they take care of the viral, infection. So we will see it, like that's what it is. That's what we have to talk about in the chapter. Now, a little bit in the history. So, Russian-born scientist, Ali Machkinov, he was looking at, let me show you. This is a larvae of starfish. It's very transparent. And what he noticed that if you take a Christmas tree, those thorns, and you poke them, they saw some cells, they started to move towards it. Means these cells, they got a signal, right, that something foreign is coming, like if you poke inside. So that was a foreign material, and these cells, they started to go and attack this thorn. So he worked on it, and he found that our animals, like blood, has some kind of cells. Now we know that these are the white blood cells, and they gather at a site where there's a foreign attack, and they take care of it by killing those cells, like foreign cells. And he also got Nobel Prize in 19. Okay, so now let's talk about, first of all, that baby is born and how natural we are eating food. Those are plants. We are eating meat. Those are animals. And how even then we are not like the moment we are eating food and we get sick. That's not the case. We call this process as a species resistance, that we are protected. Why? Because the physiological processes occurring in our body and what pathogens need, they are highly incompatible processes. So we are not compatible with one another, and incompatibility is the major reason that we don't get sick every day. Okay, for any viruses to attach to any anything have to get inside, we need these correct receptors on our cells. And our skin is without any receptors. That is a great thing. Very important thing is that skin don't have receptors. That's why we don't get HIV. Maybe those viruses are around, but even if they land on our skin, I'm not talking about the wounds or open body tissues. I'm talking about that our skin is such a good, like a protective layer. They don't have receptors. If you don't have receptors, most of the viruses cannot get in. Conditions in human body may be incompatible in terms of the pH, in terms of the temperature, like in terms of oxygen availability. Like we were talking about aerobic microorganisms, anaerobic, facultative, and deep down into our tissue, like you can't get the oxygen that easily. Maybe that's why they can't stay in our body for longer period of time. Now let's divide our body parts into 3 lines of defense system. First 2 is the part of our innate immune system. So first line of defense is external physical barriers like skin and the lining of all the systems, like your mucous membranes of respiratory system, digestive system, urinary and reproductive. Starting from your mouth to the anus, that lining comes under first line of defense. And those secretions, mucus secretions coming from those cells in those lining is first line of defense. What is the second line of defense? It's internal, composed of protective cells. These are special cells. These are blood-borne chemicals. These are the processes that these cells are going to kill your invaders or kill your pathogens. So how first line of defense is external. However, the lining is not external, but you can say that it's part of that opening, and mucus cells present in that, they take care of the pathogens. And the second one is blood-borne chemicals, blood-borne cells, and the processes these cells display. It's a first line of defense. What is the second line of defense? The third line of defense, that is our chapter 15. So together, the first 2 line of defense are called innate immune system, and they are present at birth prior to the contact with the outside the world, like where you have all these infections, right? Now, as I told you earlier, that these innate immune system works very far on short notice, whether worms are coming, whether like bacteria are coming or viruses are coming, they will take care of these pathogens by using all these means present in first line of defense and second line of defense, right? But the third line of defense, which I told you, adaptive immune system, that is specific. These are the two things, specific. And they also keep memory. And what they are mostly made up of? B cell and T cell. They have a very specific system to fight the infection and looking in such that they act more effectively upon subsequent infection with the specific strain. So, if the same strain when I was like 5 years old or 10 years old, and the same strain is affecting me again, these cells, the memory cells, come fast forward so that they can fight that infection very with more power. That's what it is. Okay, I will just skip this slide, and in the end, when we come, because right now this is the summary of our chapter. You will be lost, they are just trying to talk about skin and mucous membranes. Microbial invasion takes place. So what happens? Complement system, PRR, like what this thing happens. These processes come into play. So that's what the whole chapter is. Okay, so let's skip this one first. You guys know about the skin. It's made up of dermal layer. Like you can look at that tightly woven this layer. And this one is mostly the top layer is made up of keratin, which is like impermeable to water. And mostly pathogens cannot cross, and some cells are sloughed off every day. So pathogens or anything that is lying on our skin will be sloughed off. This is just showing the lining of our mucous membranes. And this is the goblet cells. They need mucus, and mucus helps us trapping these pathogens, mostly the columnar cells, or they have cilia also. Cilia also kind of propels the pathogens, bacteria, viruses outside. So dermis, epidermal layer, keratin, all these kind of things in the skin protect us. Now, they're just talking about this mucus lining. So starting from mouth, digestive system in the red, and everything is just like a part of your first line of defense. Okay, these are the secretions. These are non-specific chemical. They are produced by different parts of our body. So, for example, oily skin, or oil-producing cells. Lysozyme is present in our tears and saliva, although it's for a short period of time that our food stays there, but they can take care of the peptidoglycan layer of pathogens. Lactic acid, electrolytes in sweat. Sweat is also salty, and it changes the pH. So pathogens mostly will not find it very attractive for them to grow there. HCl, like very, very low pH in the stomach, like pH is 2 point something, and that's going to kill your pathogen. Bile juices, digestive juices also change the pH of the intestines, and most of the pathogens don't find it very nice for them to stay there. Semen also contains some antimicrobial chemicals. Reproductive structures in women also acidic, and that keeps most of the pathogens outside the body. So that's how they are just saying that first, like antimicrobial secretions are present, like what I told you right now. So pH in our reproductive structure, like in the intestines, physical barrier, lysozyme in tears and saliva. We have normal microbiota on our skin. We have normal microbiota in the gut, right? Flushing act of urinary system is also another way that pathogens will go away. Now, let's talk about something that we didn't mention yet, and it's about normal flora, that's what we call them, or microbiota. These are, sometimes we call it in layman language, good bacteria. Let me tell you that these bacteria are so good for us, and why? The thing is that, how do they protect us? It's a symbiotic relation. Let me tell you that, first of all, why they are there. So what are they getting from us? Symbiotic relationship is, it's a physical relationship between two microorganisms or organisms, like here microbes and humans. So what they do, they get shelter from us, they get food from us, but what do? They cover the sites, and when the pathogens come, they cannot sit there because it's already occupied by them. They consume the nutrients, and they don't let pathogens have anything left for them to multiply. Some of these, they produce like propionibacterium, they degrade lipids and make some fatty acids, which is not good for foreigner, like for these microbes. E. coli may synthesize polysyns, and they look like antibiotic, kind of. They can make antibiotics against pathogens. Lactobacillus present in our reproductive structure also keeps the pH low. So what happens, like most of other pathogens, like we go to the restroom, like, we have to go. So mostly our reproductive structure is protected by keeping the pH low in the system by two microorganisms. One is yeast, Candida, actually we call it Candida. Only women have Candida, and there is lactobacillus, which is bacteria, lactobacillus, which is mentioned here. They like a bacteria. So bacteria and yeast present, these are the two species. If there is an imbalance, let's say lactobacillus is down, yeast will go up. And sometimes women do get this opportunistic pathogen, which is Candida albicans or candidiasis. Okay, so there is also disruption of normal microflora. Let's say some people are getting antibiotics, or they are on this antibiotic therapy for a longer period of time, or some people are immunocompromised. Sometimes our own bacteria, we call it like C. diff infection or Candida like it's part of our body, like microflora, but we can get infection. And when that happens, we call these, they are not true pathogens. We call them as opportunistic pathogens. So remember this term, that what they are, pathogens. So opportunistic pathogens are the pathogens that cause the problem in your own body when you're immune compromised, or we are on antibiotics for a longer period of time. Okay, which of the following is not an example of first line of defense? What do you think? Hmm? Hello, are you guys listening?
Yeah.
Just thinking.
This is yeah.
I have to check. Who's listening?
I
Let's
want to
see.
say C.
I want to say C.
C.
C as in cat.
Yeah.
How about white blood cells? I feel like it's A, no? Because lysozyme, HCl, everything is first line of defense. The secretions of, because what is your first line? The skin, right? The lining
Yes.
of eye, so that makes lysozyme. Your saliva, that is also lining of your mouth, which is your first line of defense. And I'm saying which is not an example. So white blood cells are found in blood, and those are your second line of defense. The processes and the cells in the blood, they are linked to the second line of defense. All right, that makes sense or no?
Yes, it makes sense. Just took a minute.
Yeah, sure. Okay, before I go to the next slide, without looking at the slide, tell me, what do you think what's in your blood?
Heme.
Heme? Yeah, that's what you said?
Yes, sorry.
Yeah. And what is heme? Can you tell
Oh.
us?
I just know that our blood is made of heme and plasma if you centrifuge it.
Okay. Anybody else? although you can see there, but what do you think what the blood is made up of?
I know it has some nutrients in it.
Yes,
proteins.
definitely. So when we eat food, where does it go? It goes in the
bloodstream, and then waste as well.
Yeah, waste as well. So it carries the gases as well as, yeah, so let's see. All right, so because in this chapter we are going to talk about like the cells, which is our second line of defense, and their processes. So let's first of all understand what is in our blood. So I do have these couple of different pictures, as you can see, that when we centrifuge our blood, right away they make two layers. One is red in color, the other one is a little whitish. So this part here, very little one. And when they looked at it, that's why their name came white blood cells. And this was red, and they said, oh, it's a red blood cell. That's how they found like these are different types of cells. And in this layer also, we do have platelets. And, you can see like these cells, in a minute, we will talk about their nucleus. So usually, the nucleus of cells is circular, but they found like, under the microscope, first of all, they thought like, oh, there are 3 nuclei, or maybe 2 nuclei are here. But that was the contrast when you see under the microscope. But later they found that no, it's like the shape of the nucleus is a little bit different in these white blood cells, or, and this is an example that we also call it anucleated, because anucleated means no nucleus. This is the only cell in our body that they don't have any nucleus. Okay. Now, when they looked at this part of the blood, they found, we call it as a plasma. So plasma is without cells. It has water, it has ions, it has proteins, other nutrients, waste, gases. Everything is in this, like albumin or is present as a protein. Other complement proteins are also there. So that's the blood is made up of. Now, blood is born, like you see this process, hemophysis. So hemophysis is production of blood cells, like white blood cells, or you can say red blood cells. And they are born, like, all the red blood cells, I think they're recirculated, they die, and we make new blood cells. Even we go for the like a donation of the blood, because you can make all these cells again. So hematopoietic cells or stem cells are, as you know, like stem cells are the cells who can differentiate. But these cells, hematopoietic cells, they can differentiate only into blood cells. Not like you can't make liver cell from this one. You can maybe dictate it later with treating chemicals, but right now we call them as a multipotent cells, because they can differentiate themselves into white blood cells, red blood cells, maybe B cells, T cells, and we call them as a stem cell. So they originate from one type of cell in bone marrow, and then they differentiate, okay? And they need something that to form these new cells, we call it as a colony stimulating factor, CSF. That's how we call it. Colony stimulating factor means you have to induce that whenever you are short of these cells, like bone marrow has to be induced, and it is automatic by this factor. These cells also have different names. So erythrocytes, like these are the red blood cells. The function of red blood cell is to carry the oxygen, transport oxygen, and they have hemoglobin that binds oxygen. So these are not part of your innate immune system. These cells play only role in carrying the oxygen or transport oxygen. There are platelets, like your blood is always in liquid form and it doesn't clot. So there are some factors, we call them platelets, and thrombin, some other proteins. They are in inactive form, and only when needed, they become activated, or like they are activated, and they originate from these megakaryote. Megakaryote means their nucleus is so big, and they fragment into these small platelets, and that's what the name comes from. So platelets are something like these, fragmented cells, or they are not dead cells, but somehow they also originate from these big cells. White blood cells, we call them as leukocytes, and they play a role in host defense system. So let's have a look on the white blood cells. Further, they notice that the white blood cells or leukocytes, either they are granulated or agranulated. What is granulated means? They have granules and they kind of use them to defend the host against the pathogens. How? We will talk about that in a minute. Agranulated means they don't have granules. Another thing they notice that these granulocytes, site means cells, they have a lobed nucleus. And agranulocytes, they don't have lobed nucleus. So you can see these are granulocytes, granulocyte, and this one is a D cell, which is the A granulocyte. And these tiny dots here, these are the granules. That's why you call them granulocytes, right? And these are clear, like if you look at these macrophages, mostly they don't. So let's talk about that. So if you look at the blood composition, I do have another slide here. As you can see, the blood plasma, this one is formed element. We also call it as a formed elements, which are your mostly these cells. Here is another like picture just to talk about these cells. So one thing you notice, these are white blood cells, and there are 3 types. Actually, they use the staining. So they use basic stain. Let me tell you why the name comes. They use the acidic stain. So acidic stain, we call them acidophils. We also call them, because the dye was eosin, so the name came for acidophil. Also, you can call it eosinophils. Eosinophil is acidic dye, and acidophils and these are the same names for these cells, eosinophils. Basophils, they take the basic dye, and their granules are also different. What about the neutrophils? They stain with both dyes or both stains, So we call them as a neutrophils because they are neutral. So remember that their names, it will be easier for you guys to remember basophils. If you try to remember it without any understanding, you will get lost like, okay, basophil, oh, basic dye. Eosinophil is that acidic dye. Neutrophils, they can stain both. They are granulocyte because they have granules. So I do have a lot of room here for making different kind of questions, like what are the granulocytes, what are the agranulocytes, and which one has a lobed nucleus or so, right? And the other 2 types are monocytes and lymphocytes. As you can see, lymphocytes, they are the part of your adaptive immune system, B cells and T cells. And if you look at their nucleus, actually, it's circular. Actually, they are agranulocytes. I don't know why they are showing these colorful things here. So that may be a mistake. And I didn't pay attention that they're showing that. Pardon me for that mistake. And you can see here, like what else is present in your blood, like plasma hormone, because hormones also travel in blood. Like if they're secreted, they go from one part of your body to the second part of. They are traveling from brain to, your reproductive structure, your kidney, your blood pressure, everything is maintained. Here is another slide for you guys. So know your cells, know the blood cells. This is the stem cell. This is one stem cell. It differentiates into 3 types: erythroid, that will become erythrocyte, myeloid into white blood cells, and lymphoid cell into lymphocyte. And look at the monocyte. You have the largest in size, lobed. Look at the granules. These are the platelets. And they're just also saying platelets in clotting, basophil in inflammation, eosinophils are phagocytes, lymphocytes play a role in adaptive immune system, and all of them erythrocytes play in gas transportation. There's another one. I think these are, if you look at the percentage, right, if you go to your blood analysis, you will see that they say, oh, neutrophils should be 55 to 90% in your blood. eosinophils 1 to 3%. But anytime if you're sick with the worm infection, let me tell you that 1 to 3% will go maybe 5% or 10%. If they see more of eosinophils, means there is a parasitic infection. Neutrophils are mostly when you get sick with your bacterial infection or viral infections. These are going to be high. We also call them as a polymorphonucleosides because their nucleus has multiple morphologies. If you look at the neutrophils, if you look at this, their nucleus is really polymorphonucleosides. Basophils are very less. They play a role in allergies, and there are some cells, mast cells, they are like stable in the tissues. We call them non-motile, but they are the same thing. Like we call them the tissue basophils, and they produce histamines. People who get allergies, they produce loss of histamine because they are provoked unnecessarily, and we have to take to counteract this one allergy medicine. We call them as antihistamines. Right? So function of neutrophils and eosinophils, their major role is to phagocytize the pathogen, means eat it, in easy way, eat the capable of diaphoresis. I will talk about that, what it is. It's a process where, for example, this is your blood vessel, and let's say this is your tissue where the infection or the cut happened. These cells, these neutrophils, and they can go out, means kind of leak out of the cell. We call it diaphoresis. Okay, you will see that coming in next slide. So this is the thing. That is how they look under the microscope. If you look at the basophils, actually this is the, it's not like two nucleus, but it's a contrast. And these are the white blood cells, sorry, red blood cells. Red blood cells don't have any nucleus, so that's how they look under the microscope. Neutrophils, eosinophils. Look at the lymphocytes, they have a big nucleus, and these are monocytes. These are all agranulocytes. Monocytes later, they differentiate into, let me tell you that, macrophages. You might have heard about this. And there is another one, dendritic cells. So always remember, when you come across these two cells, their parental cell is monocyte. The monocytes later differentiate into dendritic cells and macrophages when they are in the tissues or taking part, participating in the different. I already told you that monocytes are the largest sized, right? And kidney shaped, and what they do, they differentiate in macrophages. Lymphocytes are the second most they make the antibodies, actually, the B cells. So that's why, antibodies, these antibodies. T cells target viruses, and I will talk about that later. Cancer also is fought with the help of T cells. B cells make antibodies. So these are a lot of slides, actually. Look at this one, lymphocytes and monocytes. The monocytes differentiate into these two types of cells. There's one more cell I want to talk about, natural killer cell, and you will see it's working here in innate immune system, in both immune system. How you will see that coming, like how they fight. They're non-specific, actually, they don't keep the memory, although they participate in our adaptive immune system as well, and innate immune system. So let's see how they play a role here. You will see lymphocytes in adaptive immune system. They are specific, so we will talk about them more next chapter. But dendritic cells, they are also known as sentinel cells, scouts. They engulf material and they inspect the system, and they kind of tell the adaptive immune system that there is a problem. So they are guards, actually. and they tell the problem to the B cells. These are, we call them as non-motile, like our body organs, different organs, kidneys, all of them are, there is a word I'm missing right now that is used to tell them. So kind of, they are present in these tissues like Kupffer cells in the liver, alveolar macrophages in the lungs. Oh, resident cells, resident macrophages. Resident means they just stay here, like kidney, they protect these tissues. And that's how we call them. They have different names here. And these are again, another slide just to tell. Oh, fixed macrophages. That's what I was. Because they are fixed, they don't go from one place to another. They are just found in liver, brain, lung, synovium fluid, kidney, skin, and bone. And these are the different ones, so know them. This is one more slide to talk about them, so you guys can see that. In order to understand innate immune system and adaptive immune system, there is something we have to talk about, that how cells communicate to one another. If I said that dendritic cells are acting as scouts, so how do they tell others? So let me tell you that there are some features on the cells, we call them as the surface receptors. They act as eyes and ears of the cell. Let's say this is one cell and there's another cell. How do they talk? How they talk? That talk needs the receptors. Another thing is how they need the chemicals. You will come across these names, cytokines, voices of the cell. So we do talk. So these are the eyes and ears. So how do they talk? They release something, it goes from one cell to another. From here, something is coming and they take the signal within. Okay? It's very important to understand the receptors, because the receptor is everything, like receptors in our nose. They are responsible for knowing the smell, the food is good or it's bad, the signal. So within the tissue, within the blood, within these cells, how they communicate, we need the receptors. They bind these small molecules. Sometimes these molecules are known as ligand molecules. There is something, adhesion molecules, like they help one cell, let's say this one and this one. So these molecules that allow one cell to attach to another cell. Cytokines, as I told you, these are the voices, and they diffuse to others. They are produced in very small amount, and as I told you, low concentration. They can affect locally, regionally, and systemic throughout the body. Means they can travel from one place to down to your, extremities, and that's where they can act. So let's talk a little bit about these cytokines. Cytokines have different names here, although these are all chemicals or ligand molecules. So chemokines, they help in chemotaxis of immune cells. What does that mean? So let's say this is a macrophage and something happened here. So these chemicals, when they produced here, they will move towards this chemical. We call it as a chemotaxis. It's stimulus of the chemicals. Colony stimulating factors, I already told you, needed in the bone marrow for multiplication of the stem cells into, or differentiation into, white blood cells or red blood cells. IFNs are interferons, and they control only, you have to remember, only viral infection. They don't work on your bacterial infections. Look at the name, interleukins, IL-2, IL-4, IL-6. You will come across this. These are produced, interleukins means these are produced by leukocytes, and one white blood cell talks to another white blood cell via IL. So this will produce something and they will go here and they talk to each other. And mostly you will see that coming in adaptive immune system. TNF, tumor necrosis factor that takes care of, that participate in inflammation, TNF, and apoptosis. What is apoptosis? Apoptosis is a cell death. It's a programmed, and it's a very big mechanism. It's a very complicated mechanism where they talk about apoptosis. The cells take a call that, oh, we should kill this cell, but why? They should have a reason. Otherwise, you cannot necessarily randomly, get rid of any cell of your body. So we call it as apoptosis. Some people work only on apoptosis in our body. So far, so good. Are you guys okay? Are you guys able to follow me?
Yes.
Yes.
So here in this table, you guys can see different types of chemokines, interferon alpha, beta, gamma. We will talk about this antiviral, these chemicals in a minute, and these are interleukins 1, 2, 4, 6, TNF. You guys can look at this table. So we now know we have do you guys need break or you're okay?
We can take one if everybody wants one, but other than that.
Okay, so let's talk about the next part. So now we understood that, okay, how cells talk to each other. They have receptors, they produce some chemicals. But when the pathogens come, what do they do? Like you can imagine that pathogens have entered inside the body, in the cells, in the tissues, in the blood. What happens? How do they recognize? Do they have some system? And definitely when they understood the system, they found something on this slide. It's a little bit, of the alignment. So let me tell you two things here. You have a pathogen. Pathogen is a microbe, and you have a host, right? This cell has receptors, and these receptors are used to recognize pathogen. Okay, so we call this as a pathogen recognition receptors. One term is clear. Who has it? PRR. If I ask you, PRRs are present on the host. That makes sense? How about this one? So if you have a bacterial cell, let's say this is a bacterial cell, they have a flagella, they have fimbriae, they have their nucleoid, plasmid. So this part, the flagella, the fimbriae, we call them as a pathogen associated microbial patrons or danger associated microbial patrons or pathogen associated molecular patron. That's what you call it. Anything related to the pathogen, you call it PAMP, DAMP, or MAMP, microbial associated molecular patrons, MAMP. So, two things. One, on pathogen, we call them MAMP, PMP, DMP. The second is PRR, pathogen recognition receptors. And who has it? The host has it. And these receptors, they have different receptors, means, see, I told you that receptors are eyes and ears of the cell. So what do they see? What do they see? They see lipopolysaccharides, peptidoglycan, lipoic acid, lipoproteins, flagellin, viral RNA molecules or DNA molecules of the foreign material, antigens who are coming, invading our body, right? So these are two more terms present on the pathogens. Next thing is cell, differentiation of cell and non-cell. these cells, like you can see that this is a white blood cell, the purple one, they come in contact with the self cells. They should not react, no reaction. If they start reacting, we will have autoimmune disorders. That makes sense? Have you ever heard about autoimmune disorders? So if your immune system start like attacking your own cells, that will be a big problem, and that should not happen, right? So we say that these cells do not attack these body cells, so they should not react. However, when they find or they come in contact, because it's a physical contact, right? There are a lot of things are present in your cells, in your blood system. So what happens if you come in contact with those cells? How do you react? Do they eat them? Answer is no. But if you come in contact with TMP on the microbe, these patterns, oh, this is a spike of the virus. This is a flagella. This is a fimbria of the bacteria. What are you supposed to do? Here, look at this cell. They try to engulf it, encapsulate, and then use the lysosomes. Lysosome to lyse it, the virus, the bacteria. That's how they work, white blood cells. That's the thing. Just remember these terms. I have it here. Now, these are special names, although we did talk about the receptors, special names given to these PRRs. We call them as toll-like receptors. So toll-like receptors are pathogen recognition receptors present on the cells. Macrophages, dendritic cells, cells lining sterile body sites like your reproductive structures. And we call them as a toll-like receptors. Where they are present, what do they do? Look at that. So this is the outside cell membrane of our body, like the cells. And these receptors are specific. They will detect flagellin, they will detect LPS, they will detect cell wall of bacteria. And they may be present in the inner like your organelle, right? But remember that what are the toll-like receptors? They are present on membranes. Now this membrane is on the organelle inside or outside, it doesn't matter. So you can see physical receptors. Then there is another one, node-like receptors. These are also PRRs, but in soluble form in the cytoplasm. That's how you have to know. So these are like hidden. It's like, sometimes like these cops, they are not in their original uniform. So they are undercover agents which are found in the cell. And what they are looking for, any pieces of bacterial cell wall, any flagella, any double-stranded RNA, or you can see peptidoglycan layer, anything that is going to affect the cell. That makes sense? So these are kind of undercover agents, not in the presence of like physical receptors, but they are in soluble form. And what they do, they can join these proteins in the cytoplasm and they can make this inflammasome and result in inflammation. Inflammation is just like how the fire truck comes and take care of the burning like a building. But it does damage, later we will talk about that. So far we were talking about first line of defense. And now the second part is the second line of defense means blood-borne cells, blood cells, and chemicals produced by them, and the processes. That makes our second line of defense. So far we were understanding the receptors, the blood, and now let's see. So these blood cells, which are, let me tell you this. So what they do, they have some processes like phagocytic process or non-phagocytic process to take care of the pathogen, whether it's a worm, whether it's a virus, whether it's a bacteria. And then there is a chemical defense system. It's also a process, but it is mediated with the help of these interferons that take care of viruses, right? Then complement system, inflammation. So 1, 2, and 3. Like fever is also good to a certain extent to take care of the. There is something I want to talk about, opsonization. Just remember this, you will come across when we talk about the inflammation. So opsonization is the process where, let's say, this is the pathogen. It landed into, let's say this is a pathogen, a virus or bacteria, or it's a bacteria. So what happens, the blood, like these components, they highlight, use this one, they highlight this pathogen. Like, you know how we highlight important facts in our book, they highlight this for digestion. So macrophages, white blood cells recognize them faster for digestion. So see, opsonization is the process by which a pathogen is marked for indigestion, ingestion, or destruction by a phagocyte. So let me talk about the phagocytes. What are the phagocytes? You already know what phagocytosis is. In chapter 4, we learned about bulk transport so that paramecium can eat food. It endocytizes, it eats it. Right? Similarly, there are 3 types of cells, or you can say 4: neutrophils, eosinophils, and macrophages, which is monocytes and dendritic cells. What do? They eat, they engulf, they all engulf the pathogens and digest it. Macrophages and dendritic cells, sometimes they can also show it to T lymphocytes. In adaptive immune system, you see major role of these cells in the adaptive immune system. So what they do actually, they kind of on their receptors, after digesting it, killing it, like they are showing on their hands that, oh, look what we have, we got this foreign thing and they invaded our body, so you take care of this kind of thing. They tell that message to the B cells and T cells. Eosinophils, I already told you that they take care of these parasitic infections. If they say worm-like parasites or any fungal cell, they will take care of it. There is something, neutrophils are the first responders. So their work is to respond to the first, like any foreign cell or foreign microbe, and they phagocytize it. Now let's look at the mechanism of phagocytosis. I told you that chemotaxis will be used. So let's say in your part of your body, there is something happening wrong. These cells are going to release some chemicals, and these phagocytes, neutrophils, eosinophils, and macrophages, they will run to the site. We call it chemotaxis. Right? If it's they are, like I told you, that diaphysis means they will squeeze out of the blood vessel. And let's say, sometimes we have a cut in our fingers in the kitchen, and after some time you see swelling. So what has swelling means the cells have come out and there's a swollen blood vessels. And PMPs, they recognize with the help of these TLRs, these PMPs. What they do, they fuse after engulfing it, make a phagolysosome, and they destruct it, like lyse it, break it down. How? There is oxygen-dependent respiratory burst, liberation of acids, degradative enzymes, nitric oxide. Everything is released by these cells and they kill the virus or the bacteria. And the waste goes out of the cell. We call it as an exocyte. So here, opsonization is also taking place. See, this microbe, the pathogen, is marked and it is highlighted by these complement proteins. We will talk about that in a minute. And what do you see here? This macrophage is attaching these cells, engulfing them, and they make phagolysosome. These tiny lysosomes, they fuse and they break it down and they lyse it, and exocytosis. This is another slide just to show you the same thing. Yeah, you can see phagolysosome, phagolysosome, and they destroy these bacterial cells. I like this particularly, like how macrophages are breaking. In non-phagocytic cells, rather than digesting, actually, they found that what is the role of these granules? So these granules, they kind of themselves, they kill themselves. So they release these granules, and sometimes these granules have enzymes, and they can kind of, take care of these pathogens. Rather than engulfing them, they attach to the parasite, discharge like these destructive enzymes, and then they take care of the pathogen in a non-phagocytic pathway. So just read about. There is another one, natural killer cells. They also don't destroy the pathogen by engulfing it, but they release this death package. First of all, they recognize it. They release this death package, which is made up of this, let me tell you, it's not here, but there is a granzyme, and there is a perforation, like the perforation means they make lyse the cells, the enzymes are released inside, and they take a call that, oh, we have to kill this cell, the self cell of our body cell who is infected by a virus. The second part is, so we did discuss the phagocytic and non-phagocytic, second line of defense. The second part is chemical defense system, and these are the three major systems. Let's understand what happens interferons. So interferons, alpha is made by macrophages, lymphocytes, beta by these epithelial cells, fibroblasts, T cells make gamma. What is their role? Take care of viruses. So here, let me tell you that it's like, two neighbors. These are two neighbors side by side. That's how our cells exist, right? Virus got into this cell. Now this cell can talk to this cell via IFNs. And they can get ready, this cell can get ready to face the virus. So what's happening here? This virus, you can see last time when we discussed the viral infection, what happens? The viral genome came in, they start making the proteins, they replicate, and they will assemble. This cell is going to die, right? But when there is a viral infection, I can quietly talk to my neighboring cell here, and I can stimulate, or these interferons activate. So you see, interferons are binding to these receptors on this cell. These are the receptors of the neighboring, like the door, you knock it, and that they get the signal that, oh, I'm infected and you should take care. So what they do, they stimulate their special proteins. We call them as AVPs, antiviral proteins, but they are inactive. We call them as I means inactive. And this virus will come, definitely attack me. And what I'm going to do, I'm going to activate these AVPs, antiviral proteins, and they will bind these molecules. Like you can see, these antiviral proteins bind the viral DNA, and there will be no synthesis. So you stop the production of viruses, and definitely this person here, neighboring cells, will be fine, because these viruses will go and they will start attacking all other cells. So that's how interferons. I like this system, that how beautifully cells, like it amazes me sometimes, like how this system is built up. So this is the same thing shown here, antiviral proteins. You guys can take care of this slide as well. This one, I'm going to keep it short for you guys, the complement system. So let me tell you that the complement system, the name, how it came. So let me tell you that there are approximately 30 proteins in our blood. And these proteins are known as complement protein, like C1, C2, C3, something like that. They are made up of like two parts, A and B. So they break down A and B, and A binds somebody else and complement it. B goes somewhere else, complement like with the like this part of A of the second one, A1, B1, A to A, B to B, something like that. And this we called it as a complement system. So that's how their name came. Now let me tell you that this complement system is stimulated classical pathway, lectin pathway, and alternate pathway. It means, like, sometimes what happens, let's say there are two brother and sisters, and let's say sister is working, they will say that, oh, why you are not working? I'm working so hard. Or let's say it's your roommate, and I will say, I remember like I used to say, oh, I'm doing the dishes. Why don't you cook something? Or, help each other. And in your immune system, the same thing works here, that everybody's working, macrophages are working, white blood cells are working, phagocytic interferons are working, but this system here, why they are sitting. So they are stimulated by these reactions, classical pathway. Classical pathway means if there is a rise of antibodies in your system, it's a call for these complement system that, oh, you have to active. You have to be active now because there is something happening, right? So antibody level is going up, means definitely B cells are working. Lectin pathway is that it's a non-specific reaction of host serum protein, lectin to menin. Menin is, it's a different protein in the pathogens. So when that is like somehow lectin is not activated on this pathway, is activated only when there is a binding of these two proteins. And that is a call that, oh, complement system has to wake up and take a call. Alternative pathway is when there is a highlighting of the pathogen. I mean, these complement proteins, C3b, are marking the pathogen with the highlighter. It means, oh, all the other things have to come into play, right? So let's see how it happens. Only thing you have to remember, so there is this picture of a bacteria with tiny holes in it. What these holes are, these are the purple things. So it's like pin. It's like, you have the cell membrane of bacteria or cell wall, and you pinch it with thousands of these pins. But what are these pins? These are the proteins, complement proteins. So it's like C1, C3, like, all these pathways, they make this C3 convertase enzyme. It splits C3 into A and B, and then it binds to C5 and C5b. And they stimulate 6, 7, 8, and 9, and they make this pin. We call it membrane attack. It's a complex, right? It's a complex of 9 C9 protein, C8, C7, and C6 and C5. So we call this as a, so complement system is to make membrane attack complex, we call it MAC. So that membrane complexes, these are the, I think they spend a lot of time to make these complexes, right? So here you can see diapedesis is also happening, like these are squeezing out neutrophils. So that's what you have to remember, like what these complement proteins do. Now let's talk about, very small part is left for this chapter. So let's finish it. Now they talk about the inflammation, and you guys know what inflammation is. But let me tell you that inflammation, if it's very short period of time, it's fine, but long term, it's not good. Like, if you use the fire truck to like put the water on your whole building for a long period of time, it will kill the building will be damaged. So what are the signs and symptoms of inflammation? Redness, we call it rubor, warmth, calor, swelling, and pain. So every time we have a small cut, you see that definitely the blood flow will be more, it turns red, right? And when the swelling happens, the nerve endings are kind of suppressed, and that's why we feel the pain. But how, okay, this is the acute inflammation, which is like short onset and it goes away within like few days. But chronic is long term, and sometimes, because of inflammation, these cells, they gather at the site, but they're not capable of doing the phagocytosis, these all macrophages. We call them as a giant cells or granulomas. So sometimes, people go to the doctor that I feel like it's a cancer. And later they found that it's not cancer, but some cells they gather at the site, like unnecessarily, like a lot of them, and we call them as a granuloma. What are the characteristics of leukocytes that play a role in inflammation? So see, leukocytosis means a lot of neutrophils will be released, chemicals will be released. Chemotaxis, margination means when you are flowing, the blood is flowing, they cling to the walls. We call it margination. It's like you're flowing in the river and you have to go to the shores, like to these shores. These are your shores. Because the cut is here on your skin. Blood vessel is running here. So these are bacteria here. Some cells are broken. How do you take care of the cut? That's where the inflammation plays a role. So this is, you can see margination, clinging of the neutrophils to these cells. There is endothelial cells, they kind of squeeze apart so that they can squeeze out, right? We call it diapedesis. That is the two processes taking place. And here is the inflammation. So let's say in the skin you have a cut, these are the bacteria. You have resident macrophages also taking care of them. But now you see this blood vessel is swelling, like it's expanding. And these are diapedesis, means they are going out. And also inflammatory chemicals are released as a result, because of the cut and this wound. These neutrophils, they are attracted, marginated, squeeze out, and they start eating neutrophils and macrophages. And later on, everything will be taken care. So pus is sometimes it's a combination of debris here, dead cells, and after some time you see clotting is also done and it is absorbed back into the that wound. So body takes care of it. So it's again shown here how to squeeze out. Now, as a inflammation, as I told you that minimum inflammation is fine, but if it's more, it results in that damage to the building. So apoptosis is normal cell death. Pyroptosis is as a result of inflammation. So just no difference between two types of cell death. Pyro means it's because of inflammatory response. What is a fever? Fever is because of pyrogens. What are these pyrogens? These are chemicals released by your innate immune system, cells, or working of adaptive immune system, the chemicals, and they may be dead or toxins because of the microbes. Two types of them. So there are two types of them: exogenous pyrogens, product of infectious agents. Exo means from outside. Endo means it's part of your body, like cells, like or interferons are working, interleukins are working. So these chemicals, whenever there is an infection, fever is the one that is raising the temperature of your body by the hypothalamus. So how beneficial is the fever? Because it helps in killing temperature sensitive microbes, because they cannot tolerate that temperature, raised temperature of your body. And whenever there is a high temperature, bacteria also take all the nutrition, your resident bacteria, and stimulate the immune system of your body. And that's how they take care of our pathogens. And also, whenever fever is high in your body, it also is a signal for macrophages to become large in size. They have more lysosomes. So they are activated, like your all other system is like, oh, there is a raised fever, like a temperature of your body. It stimulates all the processes of your immunity. So that's how fever plays. But I think for longer period of time, fever is not good, because then the metabolic functions of your own body are also affected. So short term, I think it's fine, but long term, it's not good. And here is the summary: your innate immune system, non-specific, first line of defense, and second line of defense, and acquired means adaptive immune system. We will see chapter 15 for the third line of defense: B cells, T cells, how do they work, how cells, they kill the cancer cells or the cells who are infected, and antibodies, how antibodies working out. Okay, which of the following innate defenses is least likely to be active during bacterial infection? So you got the bacterial infection, and which one of the following will be least active? That's what they're asking.
Is it C?
And why is it so?
Because interferons deal with viruses and not bacteria.
Very nice, excellent. Yeah, that's it. So which one of the following is not phagocytic cell?
Lymphocyte, C.
Actually, they are. So giant cell, these are the macrophages they gather, but they're not capable of eating the pathogen, or they're not. That's why they make the granule. So they gather at the site, but they don't eat. They're not capable of, digestion or like eating that pathogen or engulfing that pathogen. But you will see in chapter 15 that lymphocytes also, they can engulf the pathogen. All right, here, another one.
Is it E?
Eosinophils. So
Oh, okay.
their number will be high whenever parasitic infection. If it's a bacterial and viral infection, mostly the neutrophils will be high, but parasitic mostly and if you have allergy, basophils will be.
Okay.
Okay, so thank you for listening. Any questions?
No.